Welcome everyone, standing technician, rodent training and special training. In introduction and special training is for those of action of in hematology and estrogen training do not provide information regarding the tissue structure of cells. The practical tissue structure does not training the well how to be shining the reticular fiber into liver, which needs a special training of technique. The present and cell tissue. The identifies pigment microorganisms like bacteria, parasite, carbohydrate, lipid, mucosubstances which include a bath neutral and acidic nucleic acid mineral along with connective tissue. The special staining is essential with cellular tissue structure. Special staining is acidic crème, gram staining, chrome, specific microscopic connective tissue are biochemical compound not visible through that protein method. Protein staining is chemotachlin fusion. Hemotachlin and fusion is a standard of most used staining technique. Histopathological lab diagnosis and research purpose. The staining acid compound nucleic DNA, RNA, blue or purple. The stain page comes cytoplasm, collagen, pink or red. The staining with chemotachlin differentiation building the usual staining clearing. Special staining, the connective tissue and carbohydrate mucin staining. Muscle column high blue or green, muscle thread and nucleic black. Workflow one creation VVG used the outline elastic fiber black, collagen red background is yellow. Carbohydrate mucin staining includes identification, glycogen mucin and fungi brain management. Microchrome and pigment staining. Gram staining different bacterial gram positive purple, gram negative red. Stain relation acidic fast. Comer method silver highlight fungi problem. The staining lipid red black oil red stain. Detect iron deposit blue purple percent blue. Biotic and microscope organism tissue. Chemistry stain. Protein pigment staining. HR critical role of tissue based diagnostic research allows highly trained pathological researchers to see under microscopic tissue microbiological structure or to the presence of prevent the particular cell, even the microorganism bacteria. Routine staining refers to hemotachlin and fusion stain, underlining the structure of condition. Number of alternative staining techniques have used the information pathological research need. Prepare tissue of staining. The staining we will prepare thin section only one cell thing can be cut placed into the microscopic slide. 2 to 7 women hand supporting so that cut the every becomes neatly. Techniques used to referred to virgin section, paraffin embedded section, quickly frozen preservation, hardened heat, section microtomy or freezing camper placed on the microscope staining. The section immediately before they begin decaying and then staining. Pigment first prevent the fixate that to structure support the unfiltered pigment, the paraffin phase. The process is more time consuming creating fluorescent section, prevent a better quality strain in the most cases, and the reduced sulfonation sample can be restored almost infinitely. Paraffin section will follow fixation, prevent tissue, formaldehyde-based solution, tissue sectioning, replacing the aqueous tissue processing tissue element to infiltrate the paraffin fat. Completed organic sectioning pigment is a black wash and section cutting the source. The fine section of floodal the water bath is then picked up to place the microscope slide. Remove moisture help slide the tissue that now ready the staining. Solvent the remove the wash. Slide the stain in the dish, always the part of the staining process. The section covered the cover glass makes a preparation paramount. Macrotomy is creating a ribbon of very thin section of staining. Hemotoxylin and isocyanates staining. The section of macroscopic small intestine well defined hydronucleic polyepithelial cell and plasma cells within the lamina propria. The cytology pigment has user-friend special advantages staining only order the additional information neatly provide the related example different between two type of micro morphological similar cancer type. Botanic figure of shared play staining the within glandular epithelium in a section of small intestine. The fluid lamina propria small intestine cytoplasm plasma cells staining the extreme which correspond which will develop a quality apparatus. This autonomic convolutional dermatoid place located between the smooth muscles layer, the muscular the small intestine contains aggregation of endoplasmic and reticulum and ribosomal RNA their cytoplasm. Hemotoxicin isocyanates chemistry. H and E stain used to hemotoxicin and isocyanates. The combination used dye stain different tissue element. The basic dye purple the blue color stain in the acidic phosphorylic structure including the cell nucleus with nucleoprotein and an organism that contains RNA ribosome is rest of endoplasmic reticulum. The acidic dye typically reduces a pink chain basic acetylfolic structure with cytoplasm, cell wall and extracellular fibers. Dye organism. Chematizing extract logwood tree are perfect. Typically aluminum although blind the cell the structure. Cyclic chemotacillin, hairy chemotacillin and most popular. Bromine and fluorescein reaction fusion formed. Typical uses histological isocyanates Y which slightly yellowish and isocyanates B slightly bluish isocyanates Y most popular. Hemoglobin chemical structure carbon and OH group isocyanates Y chemical structure special staining. The trademark special staining is retinal staining other is the tissue cell structure element even the H and staining. Hypertension specific with protein RNA DNA staining the use immunohistochemistry. Now typically separated into third category now advantage stain. The clinic histological purpose of minor literally 100 speckle stain of isogen. The very mostly special staining is not automated to high-end staining. The automated instrument is a common staining. The built area of hand staining. Common special staining version trichromum. The staining includes a cytological observation. Pleural fusion blue iron. The staining used to detect identify ferric iron tissue preparation, blood match, bone marrow match, amount of ferric iron, homocysteine commonly found of marrow peeling. Common special stain, alkaline blue and PAS, the stain that are component properly path of alkaline stain. Common special staining, comor trichrome green, submoco trichroming area used stain identifies metal fibers. Microscope carefully. Microscope control stage of different step. The tiny accessibly looking the sliding naked eye. With second view under microscope, closed condenser diagram. Note the flat the background with second no covership under microscope view option condenser diagram.